Hi, my name is Ken Lasseson. This is part 6 of my microbiome lessons, which is being prepared over Christmas holidays, currently. And this lesson deals with enzyme analysis, something which I found very interesting. And let's go into it. So basically, enzymes are probably familiar to all of you. It's chemicals that causes things to change from one thing to another. And so it's pretty straightforward. Before we get into it, let's do a quick recap of what has been covered in this series of lessons. My first one was who am I, and my world or data perspective. I'm not a medical professional. I'm a data scientist. I'm also well trained in science. Been reading medical journals since I was a teenager and in enrichment science programs. The first practical lesson dealt with identifying bad apples in the barrel, bad apples being bacteria in the barrels, and talked about the randomness challenge. People are different from one person to another, from one country to another. There's no such thing as normalization, which means trying to find a bad apple in the barrel when you have 200 different varieties of apples. You're not sure which ones are good or bad. Is it the one which is dark red, or it's a bad apple, the ones that's green, etc. So there's a randomness challenge there. The next one was using PubMed studies to pick bad or missing apples. PubMed studies, a lot of them have been done for a variety of medical conditions, and they identified that they seem to find a pattern of certain bacteria being high or other bacteria being low. And from your microbiome sample, you can apply those to filter out to the ones which have a better probability of being the ones you really want to change. The next one after that is the Citizen Science Initiative, which is by looking at other people's samples and symptoms, we are able to discern patterns of certain bacteria causing certain symptoms. And using that, we again can determine bad or missing apples. By missing apples, I mean you don't have enough of a particular bacteria, or a correction. You have certain bacteria and the level of it is below ideal. You may have none of some bacteria, and we generally ignore that for a reason which in the earlier lessons we went through. The next one was dealing with end products. End products are what bacteria produce. Like, for example, it produces B6, it produces folate, etc. And from the medical literature, we attempt to identify where your production of certain vitamins or other things is below normal, and from that infer which bacteria may be causing a problem, as in which bacteria are not there enough to produce a normal amount. This lesson goes on to use enzymes. Enzymes are catalysts for the processing of chemicals from one state to next state. Some of the enzyme result in end products being produced, other of them just transitory. The future one will be looking at cake modulars, which is again a different way of looking at the data. And then the second from last is sorting the apples and developing a recipe, as in what type of protocol, what do you do to adjust your microbiome. And the last one is looking at future perspectives of where things could go, probably in the near future. There are technology changes happening, etc. So with that quick recap as to where we are, let's get on to enzymes. For enzyme, we use the Kyoto Encyclopedia of Genes and Genomes, which is a massive database which is freely available, and it's coming out of Japan, as you can guess by the name of Kyoto. And it contains basically a massive amount of information about which genes are in a variety of bacteria. And they have discovered or identified 6,100 different enzymes and the genes that produce these particular enzymes. Genes, to think of as a big collection of DNA. So these are chunks of DNA that produces certain genes. And their database currently lists some 5,700 bacteria. 5,700 sounds like a big number, but there's actually closer to 100,000 different bacteria known. So it's incomplete, but it covers all the usual populations which we will encounter, or a reasonable number of them. What happens is, as people do detailed DNA analysis of bacteria, they identify the genes. That information about which gene is in which bacteria eventually gets put into the KEGG database, and thereby we make use of it. And as to the scope of the data we're dealing with, we have 5.4 million enzymes to bacteria pairs. So we have a pretty big stack of data which we have to apply to your microbiome, which may only contain 500 types of bacteria in it, which means that it becomes an interesting case. But let's just get going into things without reflecting on the niceties. We have to be clear to understand there is no direct retail test which report on all 5,700 plus bacteria in the KEGG database. In fact, I don't even think there's a research test which does comprehensive testing. You'll get a good percentage of them, but you won't get all of them. So it means first point is we are dealing with partial knowledge. That I don't have a problem with. I'm trained in statistics. We're always dealing with partial knowledge. Some people who are looking for absolute answers may be a little bit disturbed and can nitpick their way away, and I won't bother correcting them. What we do is we compute distributions for each enzyme from all the people who have uploaded samples to us, and we sum up the number of bacteria that have a particular gene that produces a particular enzyme. So we go through all of the thousands of samples, we compute that out, and we end up with a distribution, if you wish, a bell curve, or hopefully a bell curve of what the numbers are. In doing this approach, we do ignore if one bacteria is a better producer than another one, or if the production is dependent on epigenetics or the environment. We basically put those issues aside because in general we don't have the information to be able to answer that question. So we basically say, okay, let's acknowledge we have a potential problem here, nothing to do about it, let's move on. And now that we have a distribution, we calculate what percentile ranking for each sample, for each enzyme. For example, your level producing DAO may be more than 95% of our people. That puts it in the 95th percentile. Or it may be so the 97% of people have more DAO producing enzymes. 97 becomes 3 percentiles. That is 3% of all the possible values sitting way down the bottom. When we do it, we also find that some enzymes are very seeing, and in general we ignore them. There's not much else we can do with it. That these enzymes may be produced by bacteria which we don't get reported on, or they may rarely occur, or these may be specialized enzymes which only occur in certain circumstances. Some of these enzymes are specific to different types of organisms. KEGG is not specific to human, it's across the board. So if we hit a rare enzyme, we ignore it because we don't know what to do with it rationally. Many enzymes are seen almost in every single sample, and for those we look for two types. We're looking for a low percentile and high percentile. Arbitrarily, we look for high being 97 percentile and above, and low we look at 3 percentile and below. Those need to be adjusted. But when you are dealing with 1,600 different enzymes, there's a good chance you could have highs and lows by pure randomness. So we try to reduce the amount of noise we reproduce by analysis by setting pretty high levels for high and pretty low levels for low, 97 and 3%. And our basic philosophy is when it falls into the high or the low percentile, it indicates a dysfunction or abnormality, which we deem to be a target for adjustment. For when we hit a low enzyme, we find that some probiotics happen to produce these enzymes. So what we do is, oh, you have low of these ones? Let's see if there are some probiotics which are commercially available which will produce those enzymes. If so, we suggest them, and we give you a list of them and how many different ones of the enzymes that will help. This does not fix the enzyme issue because the probiotics won't generally take up residence. It simply corrects the issue temporarily. In terms of human terms, a hope is that this is basically welfare to the microbiome, which will help it get back on its feet. So that's basically it. We just basically say, okay, if we make the right amount of enzymes available or have a lesser deficiency, perhaps things will start correcting, spinning up properly. And trying to encourage bacteria to produce the enzyme faces a bit of a problem because there are generally about 1,000 different bacteria to produce any particular enzyme. Which ones you want to encourage, which ones is the rest of your DNA compatible with. We cover these topics in our one, but basically for low enzymes, the only thing we can really suggest is either supplementation in a very rare number of cases, or taking appropriate probiotics which produces the enzymes which we are deficient in. Okay, dropping over to high enzyme. High enzyme means that we need to decrease the bacteria producing the enzymes. This is much simpler because we can identify the bacteria. What we have to do is work back from the high enzymes to the genes that produces those enzymes. From the genes that produce those enzymes, we work backwards to find which bacteria contains those. And then filter it to only those bacteria that are in your sample. So those are the bacteria you want to decrease as a whole. And generally we find that certain ones seem to be really dominant compared to other ones. So those are the ones we really want to reduce. So once we have identified the bacteria which we want to reduce, we have our bad apple, if you wish. We can use our existing suggestion engine to determine how to change things. So that then takes us to the walkthrough, which we'll start in a moment. Once you log onto the system, you have your samples, which may just be one, or like mine, many, and you have lots of choices of where to go and what to do. So a walkthrough is intended to cover the topic and show you a couple things. A good starting point is to go here and look up. Here we have enzymes. So if we click there for enzymes, you wait a few seconds, and we see all of the enzymes listed. 1,862 enzymes are available from the 5,700 officially, and these are going to be largely human-oriented enzymes. And these are enzymes which at least one of the bacteria that produces it has been seen in our uploaded samples. Now, if you take a look here, you see the names. Many of them may be slightly mysterious. You will see the bacteria, which are the enzymes are associated with, and you see what percentage of samples has at least one of the enzymes which are listed. If you go down and click on one of these entries, you'll actually go over to the KIG database, which will give you a lot of good information. It is unfortunately very technical information, so let's not dwell with that too much. It's just simply showing you how things work. Returning to our sample page, if you go under changing your microbiome, you have the enzyme outliers. Its purpose again to identify the bacteria which you have the highest, the enzyme which you are in the very top or the very bottom. So if I click that, and I will see the bacteria I have. In this particular case, I have a stack of bacteria which I am low in. That is, I don't produce enough of these. If I click on one of these, it will go and show the bacteria which produces this particular enzyme. Nice and simple. And returning back, we have a button at the bottom which is to get an aggregation. In other words, it looks up every single one of these and put everything together into one big stack. And this is what the net result is. And what we have is effectively account for the number of items involved. For example, Lactobacillus paracasi, we have 10. This one, Bacillus primaris, we have 16. That means it is producing 16 of the enzymes. This one produces 10. Other of them are producing less of the enzyme which we are low in. Okay, and if you again click view, you go over and get more information about it and can follow through ad nauseum in terms of information overload. Okay, now we also have bacteria causing high enzymes. In this case, if you click it, you get nothing. And the reason you get nothing is that there are no high bacteria in that list. So hence we have nothing to do. Okay, now let's go and take a look at a couple of other items dealing with enzymes. We'll come back to the high enzymes at the end of later on in this session. So let's just return to our sample page. And let me go and clear a few things around so you can see things better. We have multiple samples here. And we have two choices. One is you can look a whole bunch of bacteria and see if there's a common pattern. Or you can take a look at one particular example, usually your latest example, and the prior examples and see if you can see any major differences. So what I'm going to do is I'm going to go down and click a couple of my 2019 examples. And I'm doing them all from the same lab or the same processing of the FASTQ. It doesn't necessarily is the same lab. And as you can see, I think 2019 was a very busy year for me. And now I click enzymes. And the only thing in common across all of those samples, which is very common, I have something like 9 samples there. These two things were common. In other words, every single sample had low values of that. So I could click here, go through, and see with particular which probiotic helps. Interesting that at least 2 of these, Enterococcus faecalis and Clostridium putericum, are ones which I actually were regularly taking during that period of time, and they were beneficial. So that is the quick of that particular option. If you go back here, then you could have the options of, I'll go and find my 2020 examples and click the same thing. And what we find is I now have what has changed compared to my prior values. For example, I have a whole bunch of things which have gone low and one things that have gone high. This sample, this was the value. My prior samples were from 920 to 5480, etc. So this doesn't necessarily come up with the absolute extreme. What it does do is identify where your where the enzyme production have shifted away from the prior ones. So again, you can go and see if you want to increase it or decrease it as appropriately. And if you click bacteria causing high levels, nothing. We have one right here. So we have basically one particular bacteria seems to be the ones that causing my single high value, which means that that is likely one which I want to reduce. If I want to go in and click this mysterious one, we'll come in and find out what we have. We see it's a rare bacteria, and we can take a look at the numbers, and it shows on there. And although the numbers seems to be about the same, the combination with other things is what does it. And we have Bacillus being the only thing that reduces it, and which is actually in agreement with what we saw before. So I can flip back here, and when we click on the probiotics for that one, oops, sorry, we don't get any probiotics for high, only for low. So this particular one went up high was because one of our bacteria happens to relatively go up high, and as a result. Okay, so that basically is for comparison, and let's just go over, and I'm going to flip to somebody else's samples which shows a very different scenario. This is an example of a child with autism, and if we go down again, changing your microbiome enzyme outliers, we see where the enzymes outliers are. Notice we have a few. If you go down, you see that it's actually a massive list of outliers. When we click the bacteria causing these high enzyme levels, we actually don't have that many bacteria associated. What we do find is the bacteria are listed here. Some of these are recorded in medical literature as being typical for autism. That is, the numbers are recorded as being typically high. But here we have it in terms of these were identified not from nothing but the enzymes being produced. And what we have over here is we have two numbers. Of all of the bacteria here added together, this accounts for 40%. But its impact on the number of enzymes that are high is much more, is 52%. Other ones which we have, here we have, this accounts for almost 3% of the bacteria, but its impact is very small, it's only 0.6%. So we have two numbers, one indicating percentage of the bacteria which we identify. The other one is the percentage of impact on all the different enzymes, which are two slightly different concepts. For example, here we have 20% of the bacteria, but it only amounts to being 5% of the enzymes which are high are going to be traced to it. And now if we go and we click create a custom suggestion from these things, what it does do is go and create a hand-picked bacteria, which I'll click here. You can view the hand-picked bacteria, and you'll see it's the same thing here. It will show the values in the sample, and then gives you statistics about what the numbers are. For example, let's take a look at one of the main culprits, which is Uniformis. We have 170,000, and as you see, 93% of people have less than 82,000, and we are double that. We are not at the max here, but we are pretty high, so we have a very high level of that particular bacteria, which resulted in the enzyme shifts. So now we can go back and to the handpicked taxa, and now we can go over to looking at suggestions. Suggestions, basically all the bacteria we picked are the species level and below, so you can actually reduce that if you want. You have your choices of what you want to consider for suggestions. Some people, for example, won't do prescription drugs. Other people have no problems with it. We can go through and we can mark everything, or we could do just 1 or 2 things such as amino acids and supplements or diet styles, whatever. Your choice. And you basically can just click away. It will go in and compute out the things, and it will attempt to identify things which are the most likely thing to help. For example, here we have barley, walnuts, bitter melons. There we have some antibiotics, specific antibiotics done. We also have some other items saying don't avoid a slippery elm, gallic acid, a food additive, red meat, etc. The numbers here are the probability estimate of it's causing the desired change. We don't know how much change it is, so we cannot estimate how much something is going to change. What we can do is, based on a number of studies, have confidence whether or not it will cause a change. And then down below we have similar thing for probiotic mixes, and these are the ones which are suggested, things which have mixed impact. In other words, some things will improve, some things will make worse, and we don't know the magnitude of each direction. We just know it's a mixture, hence these are mixed impact. Generally they are suggested to avoid. Then we have negative impact, which is actually a very long list. These are things which will definitely push things in the wrong direction. And then we have the list of things with no impact. We now have types of food and vitamins and favorite oils which are suggested to compensate for the changes. So that all comes out of the custom bacteria suggestion there. Let me go back and just double check to make sure I had done the right button. It should be handpicked bacteria and handpicked bacteria suggestion. Yeah, design custom suggestions. So that's the result. And again, there's a lot of choices. Mix and match your own preferences for what is or isn't important. Again, for purposes of illustration for this autism one, let's go up and do a compare samples. You notice we have Thrive and Biome site. Basically, all these were done through the Thrive lab, but we also exported data to Biome site, which then went and created their own interpretation of the same raw data. Raw data, as you saw in earlier lessons, will give different results according to what computer algorithms are used to infer which bacteria are there. So let's go and let's work with Thrive for the moment. And I'll click all of them but the latest one. I'll hold off with the latest one, and I'll mark it Thrive up there for the latest one. And let's first take a look at enzymes. Enzymes, again, we have a lot of ones which are high and consistently high. There are 4 samples, and we have these occurring in 3 out of the 4. For example, this one, tryptophanase, you probably have to do a checkout, but it would imply that it probably suggests that we have low levels of tryptophan in the system. Each one of these you will need to do your own research on, as in what exactly does it do and what the impacts are. Now, if we go over and I'm going to do the comparison, and we now see what has changed from the latest examples to the prior one. For example, here we see some things have gone down, but a lot of things have gone up, and that would generally imply that things are probably getting worse. The degree of dysfunction is getting worse. For reference, my own very last samples, I was pleased to see basically that all my enzymes were normal across the board. All 1,500 of them were within the normal range. So I was very happy with that. Okay, so let's do a quick recap. Okay, in this lesson we looked at enzyme analysis, and it's part of a series looking at different ways of trying to identify bad apples or missing apples in your gut. I use the analogy of apples, which basically means bacteria, but bad apples in a barrel is somewhat of a nicer analogy. And we also have the aspect of missing apples. Missing apples, think of this: you are trying to bake a apple pie. Certain types of apples go very well in apple pie. Other becomes extremely bland. You generally want a bit of a tart apple for apple pie. Those could be the missing apples. You sent out somebody to pick up a bunch of apples. They came back, and some of them are bad apples, as in they basically may not cook very well. Others ones you may suddenly realize you really are missing this particular type of apple, which makes a big difference. So enzymes is what we're looking at. We use the KIG, the Kyoto Encyclopedia of Gene and Genomics, to do it. We look at the 6,100 different enzymes, which are across all living creatures. There's seem to be about 1,500 which are common for humans, and we're making use of those. We go and we map that to 5,700 bacteria to produce them, and we basically figure out how much of a particular enzyme is estimated to be produced by your microbiome sample. We are incomplete for the information we have. What we do is we compute, so we have apples to apples comparisons of how much enzymes is there, and we basically attempt to find high or low values, and high values is 97 percentile and above, low values is 3 percentile and below. Low enzymes, the only thing we can do is suggest probiotics that would increase it. High enzymes, what we can do is we can identify the bacteria that are causing the high enzymes, and as a result we can obtain suggestions of how to reduce those particular bacteria in a very, very targeted aspect. And that's it. So hopefully that was helpful and useful, and go to it. Enjoy, experiment. There's a lot of information there and a lot of options.